It's 8:01. Hi, should we start? Are we okay? Can everybody hear me? All right. Well, my name is Raymond Jin. I am an assistant professor in divisions of oncology and gastroenterology, and I want to extend a warm welcome to everybody to our first annual gastroesophageal cancer symposium. Before I start, I'd really like to thank organizing committee members, everybody here. I'd like to recognize two special people. Tez Akamaga, he is my research technician. He helped organize the website, the registration page. And then Chad Benson, he's outside, so he was my right-hand man through everything. So if you see Tez and Chad, please thank them in person. And of course, all our sponsors here. So I guess I was gonna just run through it, but let me introduce a spokesperson for No Stomach for Cancer. She's gonna say a few words.
Good morning, everyone. My name is Stacy Martin. I am a CDH1 mutation award winner, I guess. It's not really an award you want to win, but I had a total gastrectomy 6 years ago, so I am a patient survivor. I'm also on the board of No Stomach for Cancer. Throughout my whole journey, it's been an integral part of my education and support. I came to my first symposium at the University of Chicago in 2019, and that's the first time I met other people who are living without a stomach. And the support that this community provides is really unmatched. So I just really wanted to say thank you guys for partnering with us and hosting this. I hope you learn a lot today. If you have any questions, we have a table outside. There's a lot of literature and materials that we've provided as well. So welcome and thank you and have a great day.
Okay, thank you, Stacy. Now, the reason we're here, kind of the impact of gastroesophageal cancer. So how to start this, and I think this paper here, the study came out of one of the preeminent medical journals, American Medical Association, very recently highlighted these special early onset GI cancers. And these are cancers that occur in patients that are younger than age 50. They are the fastest growing early onset cancers, and they affect patients not just here in the United States, but worldwide, and gastric and esophageal cancers comprise a large proportion of these early onset gastric cancers. This summary of the main paper here, shown on the left, really shows some of these risk factors for early onset GI cancers. And what stood out to me is this word associated. And as a physician scientist, that really means we don't understand, okay? And really it's a lack of understanding of the pathogenesis of this disease on the basic science side, but also clinically, how we screen, how we diagnose, how we treat. So compounded with that is our lack of funding for gastric and esophageal cancers. This is a recent publication looking at NCI funding, National Cancer Institute of our United States NIH, largest funder of cancer research in the world. And they really looked at what cancers are overfunded, underfunded. Metrics are not just number of cancers, but also kind of disability that those cancers cause and death. So you can see the top of the graph on the left here, breast, lung, brain, overfunded by those metrics. On the bottom, esophageal, and the very bottom, gastric. Another metric to kind of put this in perspective is the absolute number of grants by NCI, broken down by different cancers. And you can see breast cancers per year gets about 2,000 grants from the NCI, where esophageal and stomach about 100 each. And these grants are kind of the lifeblood that keeps labs like mine going to be able to do research on gastroesophageal cancer. So that's kind of a bleak picture, but that's not kind of the message that I want to impart. And I really want to impart a message of hope. And I'm hopeful because of everybody in this room. I'm not talking to an empty room. I'm talking with people who are passionate. We have like-minded beliefs that we can find a cure for this disease. And that's kind of the message for the symposium, a message of hope. Our goals today for the symposium are threefold here. Advance knowledge collaboration, highlight emerging diagnostics, treatments, enhance patient care and support. Essentially, we're trying to unite basic science, clinical practice, patient support, because together we can not only push the science forward, but we can really transform the lives patients and families live with these diseases. So here's the agenda. So session 1 is going to be all about early detection, disease prevention. Session 2 is going to be about new treatments and research. At lunchtime, hoping that you would join us, we're going to have a meet and greet with Siteman researchers. Labs from mine, Dr. José Sáenz and Dr. Jeff Brown will discuss what research is and what we do here at Siteman. And then session 3 is going to be the patient experience at Siteman Cancer Center. It's going to be really highlighted by our tribute to Justin Hardy, who is basically the guiding inspiration for this symposium. And with that, here's some more sponsors. I would like to really start the first session, early detection and disease prevention, and introduce our first speaker, Dr. José Sáenz. He's an assistant professor in the Division of Gastroenterology. He is a physician scientist. And I've known Jose, we've gone to medical school together, we've trained in the same labs, and he's going to talk about early detection of stomach cancer in the United States when guidelines provide little guidance. So please help me introduce Jose.
Good morning. Thanks, Ray, for the introduction. I really want to thank Ray and the organizers for the opportunity to speak with you today. And as Ray mentioned, I'm a physician scientist, clinically trained in gastroenterology, but really my focus is stomach cancer. And what I wanted to briefly discuss today are some of the guidelines or the current state of screening guidelines here in the US. What are the obstacles, the limitations, and what's on the horizon? So before I start, I want to just kind of set the table, and I realize that the cliff notes to my talk are probably, you have them on your desk, but I want to set the table and place stomach cancer in a global and domestic context. And for those of you who are not aware, stomach cancer is the 5th leading cause of cancer-related deaths worldwide, with over almost 1 million cases diagnosed last year alone. And although stomach cancer incidence has been declining globally over the past 50 to 100 years, it's still a huge global problem. Shown here is a map of the prevalence of stomach cancer worldwide, and we can see certain regions with the highest incidence, in particular the Far East, so China, Japan, Korea, Eastern Europe, and Central and South America, particularly along the Andes Mountains. So I would argue that it's important to know these global patterns of stomach cancer, because in an increasingly globalized society, we as oncologists, as gastroenterologists, as physicians, are going to encounter patients from these areas. So we need to maintain a high index of suspicion. But why are these hotspots for stomach cancer? Why are these global hotspots? Well, for the past 40 years, we've known that the biggest risk factor for developing stomach cancer is chronic infection with the stomach-adapted bacterium known as Helicobacter pylori, or H. pylori. And it turns out that the countries with the highest infection rates with this bug are the ones that also happen to have the highest incidence of stomach cancer. Now, later in the talk, or later in the day, Jeff Brown will be discussing how H. pylori can lead to chronic changes in the stomach and eventually lead to stomach cancer. And I'm not going to discuss the hereditary form of stomach cancer, which our keynote speaker, Dr. Guilford, will discuss. But I did want to mention that H. pylori is a bug that has basically lived with humans as long as we've been roaming this earth. And I wanted to highlight a fascinating article in Sáenz roughly a decade ago, where they looked at the stomach of the Iceman. So for those of you who aren't aware, the Iceman was an early human who lived roughly 15,000 years ago. And he was shot in the back with an arrow and died in the Italian Alps. So his body was well preserved in the snow and ice of the Alps. So when researchers uncovered his body tens of thousands of years later, they were actually able to isolate, let me see if this, oh, there it goes. They were able to isolate DNA of H. pylori from his stomach, suggesting that H. pylori has been with us really since the dawn of humanity. Now that's stomach cancer in a global context, but how about here in the US? Well, while stomach cancer is far less common in the US than it is, for example, in the Far East, it's unfortunately often a terminal diagnosis when we do detect it. Last year alone, there were over 30,000 new cases of stomach cancer, resulting in over 10,000 deaths. And the 5-year survival rate is dismal at roughly 38%, meaning that the majority of patients diagnosed today with stomach cancer won't be alive in 5 years. And while that's sobering, I think there is reason for hope. Like with many other cancers, if we can detect stomach cancer at its earliest or localized stages, we can really significantly improve overall survival. But the problem is that here in the US, stomach cancer is often detected at late or advanced stages when treatment options are limited. And so if there's one thing that I've learned as both a gastroenterologist and a researcher studying stomach cancer for the past decade or so, it's that if we're going to improve overall survival, we need to focus our efforts on enhanced and earlier detection methods. So how do we go about doing that? Well, in my mind, this can be achieved in 3 main ways, and I'm going to highlight these and talk more about these for the remainder of the talk. But first, we need to improve and implement more targeted screening strategies. Second, we need to increase stomach cancer awareness through symposia like this one, but more importantly, as Ray mentioned, increasing funding for clinical and basic science studies. So at the end of the day, we fall down to money as being the major driver. And then finally, we need to move away from this one-size-fits-all approach and more towards a personalized approach to diagnostics and treatment. So obviously easier said than done, but I wanted to highlight how we here at WashU are making some advances towards meeting these objectives. So before I go into this, I do want to stress that stomach cancer is a preventable cancer. Like many other cancers of the GI tract, it develops over many years of chronic inflammation, usually as a result of H. pylori infection, and it progresses in a stepwise fashion through these precancerous stages, which I won't go into. But we as gastroenterologists can detect these stages endoscopically, and it allows us to kind of identify patients who are at an increased risk of developing stomach cancer. So again, in summary, the earlier that we suspect and detect it, the better off our patients will be. Problem is that we don't have sufficient data here in the US to really guide us. And so I think that we can learn quite a bit from countries in the Far East which have to deal with stomach cancer and where stomach cancer is a big problem. So I wanted to highlight a study out of Japan published this year. And what they did is they took patients who were either 40 or over, and they decided to scope them, so do an upper endoscopy, either if they develop symptoms or simply screen the general population. So regardless of symptoms, they would undergo an upper endoscopy. And what they found is that if they waited for patients to develop symptoms before they presented to their gastroenterologist, often these patients would, when they did detect stomach cancer, it was advanced stages, so stages 3 and 4. Over 90% of the cancers that they detected were these advanced stages where treatment options are limited. However, if they actually took patients who, regardless of symptoms, and simply screened them, they completely flipped the script. And now over 80% of the patients who had stomach cancer had it at an early stage, so stage 1 and 2, when you can actually resect it and treat it. Well, how about here in the US? According to the most recent guidelines from the American College of Gastroenterology, which were published just this year, they actually recommend against routine screening for stomach cancer. And when they talk about targeted screening, which I'll discuss in a second, they can't really provide any clear guidance on targeted screening approaches. So it should come as no surprise that the majority of stomach cancers diagnosed in the US are diagnosed at late stages, which essentially mirrors what was happening in that Japanese study. So who should get scoped here in the US for stomach cancer? Again, the data in this country are pretty limited, and because of that, there's no clear stomach cancer screening guidelines here in the US. But I think we've started to learn, and there's been increasing studies, that perhaps we should take a more targeted approach to screening for stomach cancer. And so I would argue that we should consider screening in those with a family history of stomach cancer, so first-degree relative, mother, father, or sibling. And we should also take into account the patient's background. As I mentioned earlier, patients who are first-generation immigrants from some of those regions that I discussed earlier, East Asia, Eastern Europe, and Central and South America, should be considered higher risk for developing stomach cancer. Now, in addition to broadening our screening options, I think that we need to overall increase stomach cancer awareness. And what does that mean? Well, as Ray just mentioned, stomach cancer is consistently underfunded in this country. And again, to rehash, I gave him this slide just to be clear. To rehash what Ray said, it doesn't take a PhD to realize that stomach cancer, highlighted here in yellow, is dead last in terms of cancer funding, really regardless of the metrics that you use. And again, stomach cancer screening is not offered and is not recommended in the US, and it falls down to money. It's not considered to be cost effective, which is a pet peeve term of mine. And then finally, if we're going to raise stomach cancer awareness, we need more studies. We need more funding for basic and clinical research studies that are going to provide adequate guidance for screening in this country. And we're starting to do that here in the US. So I just wanted to highlight we were fortunate enough to get some funds from the Foundation for Barnes-Jewish Hospital a couple years ago to start this Gastric Cancer Research Innovation Fund. And this took a multifocal approach to understanding stomach cancer and to building more personalized approaches to diagnosis and treatment. And so this was a collaborative effort. It was led by Taryn Bigley, who's an immunologist, and myself. And we've been fortunate enough just recently to receive additional funding, again from the Foundation for Barnes-Jewish Hospital, to expand these studies not only to stomach cancer, but also for the early detection of pancreatic cancer in a program known as PREVENT. And so what has this funding allowed us to do here at WashU? Well, as I mentioned earlier, if we want to enhance our ability to detect stomach cancer, we have to find better ways of detecting some of the precancerous stages that develop. And one of the biggest limitations that we face as gastroenterologists is that we visually inspect the lining of the stomach, but we often miss things. There are things that we can't detect by eye, and often these suspicious lesions might get missed. If we do detect them, it might be completely incidental or random. And so in the end, we're limited by our visual inspection. So to address this, in collaboration with a bioengineer and a close friend of mine from med school, Leo Shmulevich, we turned to thermal imaging. Using a highly sensitive camera, we're able to detect small changes in temperature and blood flow. And the idea is that we can detect literal hotspots in the stomach that we wouldn't detect by eye. Now, we're currently trying to adapt this imaging camera to an endoscope so that in real time we can see these hotspots and we could take more targeted biopsies of these suspicious lesions that otherwise would be missed. Now that's detection, but how about treatment? So again, in collaboration with Taryn's lab, we've actually been able to isolate cells from the tumors of patients with stomach cancer and then grow these cells into mini sort of stomachs, three-dimensional stomachs in the lab. And the idea is that we can try to see how these organoids respond to therapy. So you can imagine that we would know before we're starting a patient on chemotherapy, which may or may not work and would only subject that patient to toxicity, we would know if that patient's cells are likely to respond to it before we start the therapy. In essence, tailoring therapy to the patient. So the hope is that we can continue to make advances, but I just wanted to end on a cautionary note and also a call to action. And so as I've mentioned, stomach cancer has taken the lives of countless Americans, many of whom we grew up with or who our parents grew up with. And yes, John Wayne did die of stomach cancer. He overcame lung cancer, but he did die from stomach cancer. And while there's been a slow and steady decline in stomach cancer among American men in this country over the past 40 to 50 years, there's been an alarming rise in stomach cancer among white women, particularly white women under the age of 50. And this is data from Constanza Camargo's group at the NCI, and the reasons for this aren't entirely clear. In addition, as Ray mentioned, there's been a steady decline in late onset gastric cancer, so gastric cancer after the age of 50. But over the past couple of decades, there's been this uptick in early onset stomach cancer. We don't have great explanations for this. And I would argue we need more studies. So the call to action here is that we need to keep stomach cancer on our collective radars. Because at the end of the day, we're not doing this as researchers and as clinicians. We're not doing this for funding or grants. We're doing it really for the patients that we treat and the families and friends that are impacted by this disease. And so as Ray mentioned, I wanted to just highlight Justin Hardy, who was one of WashU's own. He played on their basketball team, and he, his battle with stomach cancer inspired countless others. And so I know Coach Juggin is here. I don't know if I pronounced his name correctly, but his basketball coach is here, and he'll be talking about Justin and the Hardy Strong Foundation, which was started by his family. So in conclusion, I just wanted to briefly summarize. Stomach cancer is, hopefully I've convinced you that stomach cancer is a leading cause of cancer-related deaths, and that we need to focus our screening efforts in a more targeted way and focusing on certain populations in order to increase detection. Finally, if we're going to impact overall survival, we need to raise awareness through symposia like this and through increased funding. And so before I end, I just want to acknowledge members of my lab, obviously the Gastric Cancer Research and Innovation Fund and Prevent, and Foundation for Barnes-Jewish Hospital, and some funding from the NIH. Thank you.
So we're gonna have a question and answer session at the end of this first session. So if everybody can hold their questions to the end, all the speakers, if okay, come up and we can field questions for about 15, 20 minutes. So next, I would like to introduce our keynote speaker. This is Dr. Parry Guilford. He's the research director of the Center for Clinical Research, Cancer Research at the University of Otaga, and he's also the research director for Pacific Edge Biotechnology. And his talk is going to update us on hereditary diffuse gastric cancer syndromes. So please join me in welcoming Dr. Guilford.
Well, thank you very much for the introduction and the invitation to be here. It's a great pleasure to be here, although it is 2:30 in the morning where I'm from. So if I look a bit bedraggled, that's my excuse on this occasion. So again, thank you very much for having me here, and I'm looking forward to giving you an update on what we understand of HDGC, an update on the clinical management of HDGC, and what we're doing for research to try and improve the care of patients with germline CDH1 and ctNNA1 mutations. So a quick summary for you. Parts of this talk get a little bit complex and, man in the lab kind of stuff, but every few slides I'll try and summarize what I've been going on about, so hopefully we can all keep roughly on the same page as I go through these slides. So as you all know, HDGC is a rare cancer syndrome with about 5 to 10 new cases per 100,000 births, and it's caused by CDH1 mutations and also ctNNA1 mutations. CDH1 encodes a protein called ecadherin, which I think I can make this thing go. Whoops, yeah. Put that back to there. And should go. Right. So the protein encoded by CDH1 is called ecadherin, and it's what's called a cell adhesion protein. So what it does, this big thing here is a cell and a neighboring cell, and ecadherin is a molecule which branches across between adjacent cells and holds those cells together. And on the inside of the cell, it's connected to a complex of proteins called the catenins or catenins, including the alpha-catenin, and they get joined on to the cell's structure. So all it's called the cytoskeleton, so all the features of a cell which give it its shape and its capacity to move and change. So really, ecadherin is just a bridge between neighbouring cells, linking the structure of one cell to the structure of an adjacent cell to enable a tissue to be solid and firm and kind of behave itself. The most striking hallmark of HCGC is the presence of multifocal intramucosal gastric signet ring cell carcinomas. So what these are I'll show you a photograph later on, very small signet ring cells, so small abnormal looking cells we find in the stomach, and they occur and often in quite large numbers. We've seen individuals in New Zealand with over 400 individual foci of signet ring cell carcinoma, and I've heard of a person from Houston who had over 800 independent foci in their stomachs. The lifetime risk of developing advanced diffuse gastric cancer in carriers of CDH1 mutations is really quite variable. So estimates vary from 10 to 70%, and depends on the study you look at to get an answer for what the actual penetrance is. And I'll explain why that is so variable in a slide or two. There's also about a 40% risk of women with a CDH1 mutation developing lobular breast cancer, and I'll briefly cover management of that later on as well. And it can finally be associated occasionally with cleft lip palate and certain eyelid malformations, but those are fairly rare. So just again to help us kind of get all on the same page, this is the structure of a stomach, and so this bit here is blown up from a small piece here. So this is the surface of the stomach. This is the side that your food arrives on, and there are millions and millions of these small glands across the entire stomach. So these glands have a job of producing enzymes and acid to help digest your food, but they have a fairly branched structure, and when a cancer develops, it'll start somewhere in this region in the middle here, and it will slowly grow, and until it breaches this muscle layer down here, it seemed to be fairly treatable if you can remove the stomach or remove the lesion when it's still confined to this area. It is considered to be a curable cancer. So that's where it all starts. So it's based on abnormalities in cells in these glands, and there are millions and millions of different glands in the entire stomach. So this is how we think HDGC develops. It starts off with one of these glands when the second copy of CDH1 gets inactivated. So as you know, you inherit 2 copies of CDH1 from your parents. If you have a mutation in one, that's no longer working, and then the second copy does all the work all through your development, and then at some point in your life, you can have that second copy knocked out just in one of those glands. And if that happens, you now have no copies of CDH1 which are active, and so there is no more of that protein E-cadherin, and so the cells start to behave abnormally. And what happens is when that second copy is lost in one of these glands, the cells don't always stay in place. They've lost that adhesion to neighbouring cells, and they will then divide and move out of that epithelial plane into this area called the lamina propria. And from there, it'll grow and form a signet ring cell carcinoma. So these are simply just really early stage cancers. They call them signet ring cells because if you look under a microscope, you can kind of see the cell is quite round, and the nucleus is pushed over to one side, so there's a kind of dark blob on one side, and the rest is full of a mucin, and so it kind of looks like a signet ring cell under a microscope. So as the cancer develops, you get these lesions forming. And so this is what I mentioned at the start, the hallmark of HDGC is lots of these early stage signet ring cell carcinomas, typified by these big fat round signet ring cells. The key thing about these is that they are really just normal cells in the wrong place. Sure, they look a bit unusual, but they are really just cells which they've come out of that normal epithelial plane and they've found themselves in the wrong environment and they've grown a bit, but they really aren't too abnormal. They are still largely normal cells, but they just aren't quite where they should normally be. But then in time, some of these lesions, and we don't know exactly how this occurs, but some of these early stage lesions can develop and become more aggressive. They look more aggressive, they change their shape, and they start to move quite rapidly into the underlying layers and can lead to disseminated disease. So there's a really tricky switch occurring, which we really would love to know more about, between this early stage, which is quite indolent, simply cells in the wrong place, to an aggressive phase where things are starting to get more clinically significant. So I mentioned at the start the estimates of penetrance, or the risk in a family for developing advanced diffuse gastric cancer is really, or the estimates are very variable. So when we first started to look at this in New Zealand Maori families, we estimated that it was about a 70% risk. So 7 out of 10 people who had a mutation in CDH1 were going on to develop advanced diffuse gastric cancer sometime in their lifetime, normally before they were 45. More recent studies have showed similar figures, around up to around 70%. But then the later studies, the risk has dropped down to 30, 35%, and then most recently a study from the NIH showed for some families the risk was as low as about 7%. So we've seen this really strange variation in numbers, and so a lot of people we scratch their heads saying, well, how could it be so wrong at the start and so right now, or wrong now and right then? And the answer is simply it's all to do with it's called ascertainment bias. So it's in this figure here, I'm trying to show the spectrum of the disease, where in the darkest colour in the middle, that's where there's the most severe penetrant disease, and then there are versions of the same cancer which aren't so quite severe. And if you sample, from the outside of this kind of spectrum, you'll see not so much cancer. But if you get the cases in the middle, you'll see, well, what you'll see will be a severe form of the disease. So of course, when you start to look at families with HDGC, the ones you see first of all are the ones with the most striking penetrance. So the ones we saw were families, Maori families, where there were dozens of people who had died of diffuse gastric cancer. And so they're the really obvious ones. The first ones they saw in Europe, the first ones they saw in the US, same thing. They were just really obvious, incredibly severe cases or family histories of diffuse type gastric cancer. So those are the first estimates. So not surprisingly, they had a very high risk estimate. But as you go along, you start to broaden your scanning, and you're starting to look a bit harder to try and find new families, and and those families won't have as much disease, and so the penetrance figures you get when you study them seem to be lower. So now we kind of look at the whole thing, and we realise, well, some families have very high penetrance, say 70% is real for some families. For other families, it's much lower, could be 10%. But the difficulty we have is that we cannot predict an individual's risk. So if you come from a family with high penetrance or one from low penetrance, we cannot say for sure that you do or don't have the risk that your family has shown in the past. So it's a really difficult area, and we have to be very cautious about making recommendations about individual risk. So why is this risk of advanced diffuse gastric cancer so variable? Well, two possible areas to look at. One is the number of early lesions is also very variable. So as I mentioned before, we've seen as many as 400 in New Zealand. But the odd person doesn't have any at all, although I suspect if you look really hard in a person's stomach, you probably would find them, but there's huge variation there. And so, of course, it makes sense that if someone has a lot of lesions, they have higher risk. Fewer lesions, less risk. We don't know why there is such variation in the number of lesions between individuals. It could be due to things like inflammation, H. pylori infection, varying states of tissue damage and repair, perhaps heavy metal exposure. We've got some recent data suggesting that cadmium is perhaps a risk factor for increasing the chances of getting these early stage lesions. But but certainly there's great variation in there, and that will lead to variation in an individual's likelihood of getting advanced disease. But what's, I think, also really interesting is that there's a great variation in the risk of progression of early lesions. So these T1A, early signet ring cell carcinomas, some sit there indolently, don't go anywhere, others will progress. And we don't know why that is yet. We don't know what controls that variation, but it's clear that other genes are going to be important. So other genes other than CDH1 and CDNA1, like we call them modifier genes, modify the risk of those original mutations. And so we don't know what they are yet, as I say, but it's likely that they are involved in things like immune surveillance. So as an early cancer develops, the immune system normally has a job of cruising around trying to find abnormal cells and destroying those abnormal cells. So it's quite possible that in some individuals that immune surveillance is very efficient, very effective, and those cells are destroyed and you don't get advanced cancer developing. But for other people, perhaps they don't have such a strong immune surveillance effect, and those cancers can develop and progress. So we suspect in time we will find that there are variants in different genes involved in immune function which will modify an individual's risk. But as of now, we still don't know what they are. The other really big question which always comes up with HCGC is what is its true tumour spectrum? So of course it started off being just diffuse gastric cancer, and then it quickly became apparent that there was also lobular breast cancer in these families as well. And so in fact now HCGC now really should be called diffuse gastric and lobular breast cancer syndrome. I tend not to bother just because it's too long. But so if I say HCGC, it's the same as diffuse gastric and lobular breast cancer syndrome. What is pretty clear, though, is that there are no other cancers elevated in HCGC. So there is not any great urgency to screen for other types of cancer. Where it gets a little bit complicated, I think, is that I think it's possible that germline CDH1 mutations may affect the look of or the behaviour of some types of sporadic cancer. And so, for example, a pancreatic cancer may be developing a sporadic cancer, pancreatic cancer may be developing slowly, but if it's combined with a germline CDH1 mutation, that may change its rate of progression, it may change the way it looks. So there's a chance that what are rare cancers could be, their appearance could be altered by the presence of a CDH1 mutation, but I don't think CDH1 is driving the formation of any cancers other than diffuse gastric cancer and lobular breast cancer. Another big question that people will always ask me is, so if E-cadherin is a protein which is expressed in all epithelial tissues, so that's all the most of your internal organs expressing E-cadherin, why do we just see diffuse gastric cancer and lobular breast cancer in families with these mutations? Well, I still don't know the answer to that question, but it's likely to be something like some compensation in other tissues for E-cadherin loss. So E-cadherin is one of a family of fairly similar proteins, and we suspect that in tissues other than gastric and breast, then other cadherins can be turned on to compensate for the loss of E-cadherin. So that's one theory. It could also be due to differences in immune surveillance, so perhaps immune surveillance is not as good in stomach and breast as it is in other tissues. Or it might be something to do with how stable that second copy of CDH1 is. I mentioned you got your one from your mum, one from your dad. The second one has to get knocked down or turned off somehow for the disease to develop. So perhaps in some tissues other than gastric and breast, that second copy is more stably expressed. It's stronger and it holds on there and it keeps things normal. These are all things we still have to understand. So to summarise the first few points there, one is that individual risk for advanced EGC is highly variable between families and within families, and we currently cannot predict an individual's risk. Family history does provide some guidance, but you shouldn't rely on it. So if you have a really low family history of diffuse gastric cancer, that makes you think, well, yeah, my risks are probably lower, but you cannot simply rely on that alone. A lot of people also tell me they don't know their family history. They're not in touch with their family. And so I think if you're in doubt what your family history is, I think you should work on a lifetime risk of around about 40% to give you a reasonable estimate of what your risk might be. DGC and lobular breast cancer are the only known cancers with higher risk, and I don't think there's any need for additional surveillance of other organs, but people should be really diligent in following national surveillance guidelines for their care. And then finally, for this first summary, take care of yourself. It's really obvious. Watch your diet, don't smoke, exercise lots. Smoking, vaping, you really shouldn't do either of those things because they probably gives you a high dose of cadmium, which, as I mentioned earlier, I think we're quite concerned that cadmium may increase risk. So keep away from some of those things and just lead a good, healthy lifestyle. Right, so we think there are around about, I don't know, it's very hard to know for sure now how many HDGC families there are around the world, but we're talking in the thousands now. There's a database called ClinVar where there's 735 different mutations are listed on there. So there's probably at least, I think, 2,000 families around the world with mutations in this gene. Most of these mutations in fact, all of them are called truncating, which means they just knock out the function of the gene entirely. So they're not activating the mutation, the gene, they're knocking the gene's function out. There are very few, if any, missense mutations which are pathogenic, and there are no major hotspots. So you can have mutations across the entire gene and they will still have the same effect of destroying the function of that gene. There is one really interesting large founder effect, which refers to a very, very old mutation, and it's been seen in Portugal, where initially they saw 4 or 5 families in Portugal who seemed to be unrelated in northern Portugal, and they thought they were unrelated. They found that they had the same mutation when they sequenced them, their DNA. And it turns out they were able to kind of take it all back and found they had a common ancestor dating back to the 16th century who had that mutation that's now spread across large parts of Portugal and also Western Europe as well. And there is very little correlation between, if any correlation, between the location of the mutation, whether it occurs at the start of the gene or the middle of the gene or the end of the gene, and how the disease is expressed in people. So if you look at your own mutation and it's different to somebody else's, chances are it has no real effect. They're just mutations which knock out the function of the gene. ctNNA1, the second gene involved in HDGC, there's still quite a few mutations reported in ClinVar, but not so many families have been described in the literature. Only about 15 or 16 that I've seen recently. And so we still don't know nearly as much about cDNA1 as we do about about CDH1-driven cancers. There's a suggestion that the risk of DGC is a little bit lower in ctNNA1 carriers, but it's still really pretty comparable. So for now, I think we just assume that ctNNA1 will mirror the behavior of CDH1. Right, now some really exciting pathology. I know when people love put up the pink slides of pathology, but this is a bit about pathology and a wee bit more about the life history. I touched on it before, but I'll go into a bit more detail on the life history of what we understand about how this disease develops. We think the primary defect in HDGC is what's called impaired cell polarity and spindle orientation. And what that means is, as I mentioned at the start, is that when cells divide, so this is what's called the basement membrane, the cells the membrane that cells sit on, and here's a couple of cells joined together by ECAD heron. When they normally divide, they divide in that same plane. Okay, so you keep things neatly in a single layer of cells. That's the normal situation. When you lose ECAD heron, or alpha-catenin, so the ctNNA1 protein, those cells no longer hold on to each other. They lose their sense of direction. They don't know which way is up or which way is down. And so when they divide, they will cause cells to go in the wrong direction, either up or down. And that means cells just end up in the wrong place. So here's an example. This is actually from one of our mouse models, but here's an example of that. Here's a normal gland around here, and here is a cell dividing out of the plane. It's kind of just in the wrong place. And here's another example of that, of those organoid things we heard a little bit before about, and a cross-section through an organoid, and these red cells are cells which have lost ECAD heron expression, and you see they're kind of pushing out of the normal neat plane, the kind of green plane. You see these cells without ECAD heron just pushing into the wrong place. So the key point here is, as I mentioned a moment ago, these early cells which form those signet ring cell carcinomas, the early stage lesions, they're really normal. They aren't aggressive cancer cells as such. They are cells which have just found themselves in the wrong place. Okay, and that's why we're pretty confident that many of the early stage cancers that we see are not clinically threatening. This is a really intriguing slide that we came up with a while ago. And let's just go through it. So what this is the number. This is from one of our big Maori families in New Zealand. So they're all related. They all have the same mutation, and they all, and about 15 or 20 of them, they had a total gastrectomy, and we counted the number of early stage lesions in their stomachs across the entire stomach. So you see up, one as high as 500 up there, but most of them below 300, but still lots and lots of these small foci of cancer in their stomachs. And we plotted that against age. And you know what you would expect to see? You would expect to see it going like this, the graph like this, wouldn't you? So as these cancers develop, you would think they would accumulate, they would stay, and the numbers would increase over time. That's not what you see. It's really flat, almost kind of going down. So it just doesn't make sense. We assumed that if a signet ring cell carcinoma would form, it would stay, it would live, and eventually it might grow. And as more developed, those numbers would increase. And so you would expect with age to get more of these lesions occurring, but you don't see that. And that, to me, suggests that many of these lesions are transient. They come, they grow for a little while, and then they die. And we don't know why that might be, but it might depend on the true cell of origin. Was it from a stem cell? Was it from a progenitor cell? And that might determine whether it lives or dies. But clearly, a number of these early lesions will not last. They will just flourish for a small time, and then they'll disappear again. But why it's difficult for us is that the progression to a more aggressive disease is unpredictable. I cannot for sure point to one and say, aha, that's the one which is going to turn nasty, or that one will never turn nasty. But we do know that many of them, most of them are indolent initially, and many are transient and will just be lost. But as I say, we've got this concern that sometimes they will go from the nice, big fat round signet ring cells to a more aggressive look. So, the cells get really unusual shapes. The nuclei gets a bit strange looking. So we get that more aggressive motile shape, and we just don't really understand what switches it over. It might be, as I mentioned a moment ago, depending on the cell of origin. Is it a true stem cell or progenitor cell, where the stem cells you think would last forever? It could be some kind of signaling from the neighbouring stromal cells, the fibroblasts, the muscle. It could be differences in immune surveillance, or it might be just simply suggest the need for additional mutations required to turn it from being an indolent lesion to an aggressive lesion. But as of yet, we haven't found those mutations, although quite a few people do have additional P53 mutations in their more advanced tumours, so perhaps it's one of the genes that does help push it along. So some key points from that section there. A family's exact mutation tells us very little about risk. So they behave roughly all the same. So if you have a mutation different to the person beside you, chances are your disease will behave the same. Most early stage lesions are just normal cells in the wrong place. So they're indolent carcinomas. They are not in themselves threatening. However, there are poorly understood triggers which can make some of them aggressive. So the management of HTGC is, initially it was really just very, very strong recommendation. If you have a CDH1 mutation, have a prophylactic total gastrectomy sometime in your 20s. And if you did not want to do that, then it was recommended that you had surveillance with lots of random biopsies taken. And if an early lesion was found, then you would be recommended to go and have a total gastrectomy. And in some ways, all that was about, if someone had surveillance, it was all about just trying to get some proof of disease so they could justify going ahead to have a gastrectomy. So it was, at that time, we just thought all these early lesions were all aggressive, and therefore, if anyone was found, then that individual should have a total gastrectomy. Today, it's kind of softened quite a lot. So the NCCN guidelines, they say that carriers should be offered either surveillance or gastrectomy, and this decision should be based on multidisciplinary discussion, taking into account patient preference. So, easing back from the just get a gastrectomy. It's the same with the IGCLC guidelines, which they've been making guidelines since 1999. We haven't got the latest guidelines yet. They're still in preparation, but we're pretty sure that what they'll show is, again, more of a balance between gastrectomy and surveillance for people who have the mutations in these genes. And there will be probably a greater emphasis on targeted biopsies rather than random biopsies. So trying to find evidence of lesions which look like they may be more aggressive. So this role for surveillance is going to get, I think, really important, where rather than trying to find any signet ring cell carcinoma, any early stage lesion, we'll be trying to find one which shows evidence of aggressive behaviour. We see that a little bit in this slide here. So this is a classic, up here you can see kind of like a pale lesion up here. And when you look at the cancer underneath it, it's just a classic signet ring cell carcinoma, not doing very much at all, a classic indolent looking lesion. Down here we've got a case where on endoscopy you see a more unusual surface. There's more erosive lesion occurring there, and when you look underneath the cancer, it's one of the more aggressive types. So we're starting to get insight, and this is work being led by the group in the Netherlands, Chella van der Post and her colleagues, and they're really starting to get a good insight into how the look of a lesion with an endoscope correlates with the actual disease underneath it all. So to kind of summarise that, the effort is really very much about trying to see lesions which are going to be really aggressive and do definitely require gastrectomy, whereas others where they may be more indolent and you can perhaps leave people alone for a bit longer. So this is trying to just kind of summarise that, so the balance is changing. So it used to be all about prophylactic gastrectomy, surveillance of people really weren't very keen, lots of random biopsies and targeted biopsies, to now where we're moving to a state where surveillance should very much be considered by individuals who have a mutation in one of these genes, And the emphasis there will be on targeted biopsies rather than random biopsies. But people, of course, should still consider, and for many it's the right decision to do, is to go straight for a prophylactic gastrectomy. Just one slide on, I know it's a gastric cancer meeting, but we can't talk about HCGC and ignore breast cancer. So the breast cancer risk is really very well managed by surveillance, although women can opt for a prophylactic mastectomy. And the surveillance consists of clinical breast exam every year, MRI from about 30 years of age, or ultrasound if you haven't got access or don't want to have an MRI, and mammograms from 40 years. So to summarise those points, around decision making, I think for people who have mutations in CDH1, I think it's reasonable that carriers should now consider surveillance as being a reasonable option. But it must be carried out in an expert centre, a centre which is really familiar with HCGC and sees a lot of these people. So they have to be able to get good at recognising the abnormal lesions. If 1 or 2 indolent type early lesions are found on biopsy, it does not justify immediate surgery. So you don't go, eureka, we've got cancer, off you go to have your stomach out. If the pathology of those early lesions looks indolent, then the people can keep their stomachs for a bit longer if they wish to. They might be best to have more frequent endoscopy, but it is not a reason to go straight to surgery if you find 1 or 2 small indolent lesions. But if those lesions look aggressive, their histology looks aggressive, or there's lots of them, or they're really big, then yeah, go straight and have your stomach removed. A very, very important thing for carriers to think about is their role in the decision-making process. It's so important that people who carry mutations in this gene are really in charge of making the call whether they have a gastrectomy or not. The decision often comes down to an individual's appetite for risk. So if you have a mutation in CDH1 or CDNA1 and it's really worrying you, you don't feel relaxed in your life. You feel every bit of stomach pain, thinks, oh no, here's a cancer growing now. This is no fun. I don't want to wait. I'm too scared to wait for my next endoscopy. It's just like no good. Well, then if that's how you feel, then you have a gastrectomy without doubt. You just go straight for it. It's the right thing to do. But if you are a little bit more prone to taking risks, then you can go, I understand my risks of getting advanced disease are quite low. I'm okay with that. I've told someone the other day I have a CDH1 carrier in my hometown who's a fireman, and he's still got his stomach. He gets surveillance, and when I explained to him the risks that he was facing by keeping his stomach, he kind of laughed. He said, "Look, mate, I run into flaming buildings every day. I'm not. I don't care about this slightly elevated risk of diffuse gastric cancer." So it comes down to individuals, but no one is wrong in their decision. If someone wants to have a gastrectomy, that is a great thing to do, and they should go and do it. But if someone wants to try and hold on for a bit longer, perhaps delay surgery until they're through a certain life stage, then that's absolutely fine too, as long as they're getting this surveillance in an expert centre who does a really good job of that surveillance. Finally, my final note is a note of warning, be cautious. One thing we've seen in New Zealand is, we've been looking at families now for 30 years, and we're now into a new generation of kids coming through. When we first found the mutations in the first family, everyone was running down to have surgery. They'd seen their mother die, they'd seen brothers and sisters die, they'd all had dozens of cousins who had died, and so it was all around them, this death from this disease. And so the chance to have a gastrectomy and get on with their life was amazing. They all took it really fast. We've got this new generation now who haven't seen death, or very little death. And so their view on the whole thing is a bit different. Sure, they know the stories. Sure, they get told what they should go and have a gastrectomy, but they kind of look at it and go, "Oh, I'm fine, and no, my mates have died, and I don't quite get it. Well, I do get it, but I don't feel the urgency." And so it's difficult. So we're finding now that the young ones are kind of a bit lackadaisical and not so excited about going to get their stomachs removed, and that's something we have to kind of really work with. We very, very sadly we lost a young man just last week, 35 years of age, who declined to be tested. His mother had a gastrectomy and she was fine, but he had thought, no, I have no interest in this. and he did not get tested, and he developed diffuse gastric cancer and passed away last week. So it can still happen, and I think, as we go along and manage families well through surgery and good surveillance, we tend to kind of forget it is dangerous. We have to be careful, we have to be vigilant. So if you do carry a mutation, you just got to make sure you either go for a gastrectomy or make sure you get really good surveillance. Right, so what are we doing in the lab to try and reduce the risk of disease or advanced disease for people? We're trying to develop drugs for the chemoprevention of HDGC, which is the intention to delay or provide an alternative to gastrectomy. And the approach is to try and target weaknesses or vulnerabilities in CDH1 mutant cells, and then to get some drugs to destroy those cells based on what those vulnerabilities might be. So as a tumour, here's a normal gland, here's one with an early stage cancer, the more indolent type, here's a more aggressive one here. What we want to try and do is target these early stage lesions before they have a chance to transition to that more aggressive type. So the question we first ask ourselves is HDGC actually amenable to chemoprevention? Well, we think it is for a number of reasons. One, it's most of these lesions are really small, under a millimetre in diameter. So and they have not much of a stromal barrier, which means there's not lots of other cells sitting there blocking the way for treatments to get in and immune cells to get in there. They don't have what's called a hypoxic core, so the middle, they're so small, the middle of the tumours, they've still got a good supply of oxygen to it all, which means those cells tend to be a bit more responsive to treatment than cells which have a kind of a hypoxic core to them. They all have the same mutation. There's been no time for other mutations to occur, so they're all really, really similar. It's not a heterogeneous population. And as I mentioned a number of times, they are still fairly indolent, and so there's low clinical urgency. Initially at least. So what we started off doing was developing various cell lines and preclinical models of cells with CDH1 and cells without CDH1. And we also developed these things called organoids. We heard about those earlier, these little stomachs which can be used to kind of mimic the disease. And so here's an example. I showed this one earlier on, but this is a cross-section through a gastric organoid from our mouse model. So just think of like a tennis ball just cut in half. The green is the ECAD heron being produced by that normal organoid. And then down here we've knocked out the CDH1 gene and turned on a red dye at the same time. And what you see is when the ECAD heron is gone, as I showed you earlier, these cells which have lost ECAD heron start to kind of go in the wrong direction. They go that way or they go this way, and it's just lost their sense of which way is up and which way is down. So very, very useful model. And we've used these models to do RNAi screens. We've done lots of drug screens. We've done lots of sequencing to try and understand how these two cell types differ. One's with ECAD heron expressed and without ECAD heron. How do they differ? And if they differ, what can we use to kind of target that weakness? And what we saw, and I won't go into too much detail on the slide, but what we saw not only is the CDH1 mutant cells, they have abnormal cell adhesion, but their organisation of the cytoskeleton is abnormal, and they've got really disrupted interactions with the surrounding extracellular matrix. So the stuff that the cells sit in, that is a matrix which signals into the cells quite a lot to tell them how to behave. That interaction between the matrix and the cell itself is really unusual, sending unusual survival signals into the inside of the cell. So we see here quite a few things that we could possibly target, and we also see evidence for altered immune surveillance. So again, we've got a few different ideas what we can use to attack these early lesions. So very briefly, what we're focusing on three areas. One is a thing called the DDR2 SARC AKT FAC axis. So this is really just a whole pile of genes and proteins which interact with that matrix around the cells. We think those are really, really useful targets to go for. We know that the membrane is around the cell is not well organised in the ECAD heron negative cells, and so so cholesterol, sphingolipid synthesis pathways are also useful targets for us. And the immune surveillance is abnormal, so we're currently looking at drugs which call the immunotherapy type drugs, which help us increase the surveillance of the immune system on these early cells. And we're taking these 3 different approaches and putting them across our mouse model. So we've developed, it's taken us years to make this mouse model. And what it is a mouse where we can give it a drug to induce the cancer and induce the deletion of CDH1 in those mice, and that will induce the cancer make this cancer start to form. And that gives us a really, really good model for testing drugs to then take on to human clinical trials. I'll give you a couple, a bit more detail on that mouse. This is a mouse stomach here, and this is the tumour down in the entrum of the stomach, a big gastric cancer there. This is a section through the stomach. The brown colour there shows in a normal stomach, lots of ecadherin and brown there, and when you have knockout of the CDH1 gene, also with p53 sometimes as well, you find loss of ecadherin in these lesions. Here's one of the tumours developing. It's classic kind of advanced cancer here with lots of fibroblasts produced, lots of collagen produced. So it really does recapitulate human disease really well. So we're very happy that we have a model which is extremely useful for testing drugs on. And it gets cancer, really quite fast. So they get in situ cancers, little, really tiny lesions within 4 weeks. Stage T1 in about 2 months, and stage 3 in 9 months. So it's CDH1 alone. But if we add in an extra p53 mutation, we can get tumours, stage 3 tumours, in 3 months. Now this sounds all a bit weird to you guys, but it's really important to us because it means we can do experiments in a reasonable time frame. A lot of these mouse models take a long time to develop cancer. You're waiting a year to get cancer. We can get cancer developing in 3 months, so we can test our drugs in fairly short experiments. And when funding is tight for research, that's very, very important. We can get our costs way down, and we get way better data because our model is aggressive. And also, we think by working on an aggressive model, we've got the best chance of getting a drug which will be useful in humans. So, key points to summarise that section. We looked at cell line models with and without CDH1 mutations, and we've found a number of vulnerabilities in the CDH1 mutant cells which you can then target with drugs, either single drugs alone, or we're working on lots of drug combinations too. And we've developed this fancy engineered mouse, which is a really good model for the disease. I'll give you a couple of examples of promising drugs that we're looking at. So these are AKT inhibitors, which both are in clinical use, although we can't get hold of the mirinsertib very easily yet, but certainly capivasertib is in clinical use. So let's look at how it's gone in the mice. So this is mirinsertib in mice. The cancers were induced for 2 weeks, and then they gave them drug for 4 weeks, and then we looked at their stomachs. What we saw compared to the control mice was about a halving of the number of tumours that were present, of T1A tumours that were present. And when you look at the size of those tumours, they were really, really small tumours compared to the wild-type tumours. So clearly this AKT inhibitor, mirinsertib, is doing a really good job at putting the brakes on and killing a lot of these early-stage cancers. The second drug, capivasertib, it's the same story, not as impressive figures. It's about a 40% knockdown in number of PT1As, but they are smaller, and so again, this other AKT inhibitor is doing a pretty good job at holding the cancer as well, but although not quite as impressive as this. But it tells us that we're on the right track, that these AKT inhibitors are really useful, and they'll form the basis of many of our proposed treatments in the coming few years. How to test them into humans is a big deal, and so we are very keen to get away from our mice as soon as possible. As soon as we get proof of principle that that a drug is useful in mice, we're going to move on to human studies as soon as we possibly can, funding allowing. And we'll be doing those studies in collaboration with clinicians around the place, but particularly with Jeremy Davis in Baltimore at University of Maryland. But the way to study these cancers it's really quite difficult to study the effect of chemoprevention because we cannot see these cancers very easily. And so what we're going to have to do is, for our first studies, are going to have to be ones in the stage 1 trial design, where we get people who have a mutation in the CDH1 who wish to have a gastrectomy. They've already made up their minds they want to have their stomachs removed. and we will give them surveillance, and then they have a positive biopsy, then they will go on to get drug treatment for a period of time before they give us their stomachs. So they are destined to have their surgery done, and we will give them drug treatment in a window prior to their surgery. Then we can get their entire stomachs, and we can have a good look at their stomachs under the microscope and see if there are any cancers there. Can we see a sign of those cancer cells dying? Is the drug getting in there? Can we see inhibition of all the right pathways in those samples? So it's a way for us to look at those stomachs and get a sense for how well the drug is working, and do that in a very detailed way. And then once we get confidence that our drug is done well in this stage 1 trial, then we move into the more exciting studies where we'll be getting people who intend to try and hold on to their stomachs for longer. And it'll be the same kind of idea. People will receive, and will have surveillance, and they'll receive drug, and that will cycle on for as long as we feel confident the drug is doing what it should be doing. But if the person loses interest and it's too difficult, then of course they can move to gastrectomy. So it's a stage development for this shift from lab models into human clinical trials, but we think this is the only way we can do it. Until we find a new way to visualize the tumours, we have to do it in this more conservative manner. One great concern that we've always had with this is that we're going to give people some reasonably serious drugs, and we know that compliance will fall off if we're giving people drugs frequently which are really hard to take. So we're not doing many people favours if they can hold their stomachs but they feel awful. So what we wanted to do is try and deliver drugs straight to the stomach and not systemically through the whole body. And if we do that effectively, then we can really reduce the side effects that people will experience. And the way we've approached that problem is with amazing work from our chemists, Jess Fairhall and Alan Gamble. They've made a stomach delivery system for us, which is a mucoadhesive polymer, kind of like, a bit like Gaviscon. I think it's very similar to Gaviscon. So this is gloopy stuff which sticks to the lining of your stomach. But what Jess has done is she's taken that polymer, which sticks to the stomach lining, and used a acid labile linker to link on our drug of interest. So it might be the AKT inhibitors, which she's building at the moment. So what that means is our drug is attached to this sticky stuff which sticks to the stomach lining by a chemical linker which degrades under acid conditions. So in the stomach, the acid will go right out. I can break that linkage, and it will release the drug right on the stomach surface. If the mix, the gloopy mix and the drug passes through to the rest of your GI tract, the drug does not come off the polymer. It stays inactive, and you get way less uptake of drug into other organs. So we've done one look at this with one drug so far, a drug called dasatinib, and we saw about a 70% reduction of drug concentration in organs other than the stomach. So we think we're onto a really good method to give a good dose of drug to the stomach and leaving the other organs with much lower doses. So what we envisage in time will be a kind of the way to care for people with CH1 mutations through surveillance will be for them to come in annually for their normal endoscopy, and then at the end of that endoscopy and the usual biopsy's been taken, we would spray on to the lining of the stomach this mucoadhesive polymer attached to our drug of interest. It might be an AKT inhibitor, and also we've got methylene blue will be attached to that as well, and so will be a visible dye getting sprayed onto the stomach surface. So that's all kind of a normal process to be done. And so what we think we'll better get is like a routine where people come in every year, endoscopy, look around, try and find any lesions, take some biopsies, and when before the endoscopist pulls out of it, he or she will spray the stomach lining with this drug linked to this gloopy polymer, which will stick to the surface of the stomach, and the drug will be released and kill any remaining early stage lesions. So to summarise those final points and my last points, AKT inhibitors can block tumour development in a really aggressive mouse model of HDGC. Clinical trials are getting really close. I'm going to Baltimore on Sunday to talk to Jeremy Davis from the University of Maryland about getting our first trial started using one of these AKT inhibitors. We're working really hard and have got a proof of principle of this new way to deliver drugs straight to the stomach, so we'll get way fewer drug side effects in time. And we see future surveillance being a combination of routine endoscopy combined with the routine application of this drug to the stomach lining. So finally, just to thank everyone. As you all know, I don't do any work. I sit in my office and worry about things, and these are the people who do all the work in the lab and in our health clinics around the country. The Maori family we worked with from the very start, Mabel and Erin and Pauline Harawira. And of course, I must thank Karen Choggin-Scriber. I know many of you knew her, and she is deeply missed, and I personally very much wish she was with us still. So thank you for your attention. I'll be taking questions later, I think. Yeah, thank you.
Thank you, Dr. Guilford. I'd like to introduce our next speaker, Dr. Jeffrey Brown, Assistant Professor in the Division of Gastroenterology, another physician scientist. He's going to talk about these precancerous metaplasias now.
Well, thank you, Ray, the organizers, No Stomach for Cancer, for putting this on. My only gripe is that you put me after a wonderful talk from Parry, our keynote speaker, and you asked me to talk about stuff underneath the microscope that most people in this room haven't looked at since high school when they looked at onion skins and what have you. But either way, I'm going to try my best, and hopefully you can follow along. All right, so no relevant financial disclosures. So unlike Parry's talk, which focused on diffuse type cancers, the metaplasias that I'll discuss on that evolve as a precursor to cancer really fall more in this intestinal type of gastric cancer. And the reason is because intestinal type is much more closely correlated with H. pylori, which causes a very prolonged period of chronic inflammation over the course of decades that allows these lesions to evolve. And the sequence of events, Jose had briefly talked about this, is something called the Correa cascade. So this is what a normal stomach looks like. I won't get into all the details of it. But once you get infected, the initial stage is you get a chronic gastritis, which itis just means inflammation. So you get an invasion of a lot of immune systems immune cells that just cause inflammation. But if this continues over a long period of time, then the stomach will eventually atrophy. And atrophy, just like as muscles atrophy or anything else, is a loss of certain things. In the stomach, this is generally seen by loss of the acid-secreting parietal cells. And generally, this over here is a pretty reversible process. There's some question whether gastric atrophy can truly go back to normal. But if you treat H. pylori, get rid of the bug, you can probably go back to normal stomach here. At this point, though, if the inflammation in some people continues, then you actually change the stomach. And you go into an intestinal metaplasia. And I'll explain exactly what metaplasia is, but it's similar to what Parry had said. It's normal cells in the wrong place. And this is a precursor lesion that is important because as I'll try to argue at the end, is there certain types of these metaplasias that have an exceedingly high risk to progress to cancer. And if we can identify those people, then we identify the people that we should probably survey. And there are certain types of intestinal metaplasia that are pretty benign that have a lower risk. So I'll stop here with most of my stuff. I'll just say that this is the precursor lesion to dysplasia and eventually cancer, which hopefully we can identify people here so we can identify these at an early stage when they're curable, not when it's too late. So what is metaplasia? So this is the definition of metaplasia. It's a change in which a mature cell type traditionally found in an organ is replaced by a mature cell type normally found somewhere else. And I underline mature and normally because this is what discriminates it from dysplasia and cancer. Because people don't normally have cancer elsewhere in the body. You can have these other intestinal-like cells elsewhere in the body, and then they move to the stomach. And that's what really metaplasia is. So, I'll point out this later, but you can see all these little round cells, these little goblet cells, these mucus-secreting cells. These are normally found in the intestine and don't have any business being in the stomach or in the esophagus and Barrett's esophagus. This is what they call with metaplasia. And metaplasia is not just seen in the stomach. This is seen in a lot of other things. If someone chronically smokes, you can have metaplasias of the lungs. I think the easiest one that we can see is metaplasia of the esophagus. This is with chronic acid and likely bile acid reflux, because the change is so dramatic. So usually you have a squamous epithelium, which is what covers your skin. And then this changes to a more columnar thing, which lines the GI tract or your lips or anus and that kind of stuff. But you can see the dramatic change. So this is metaplasia. This is what it's supposed to look like. Similarly, this gets cell types that are typically found in the intestine, goblet cells, and the mucus secreting cells. But the same thing happens in the stomach here. And the reason why I really like this slide is that this shows that all metaplasias aren't the same. This is what a normal stomach looks like on a biopsy. This is a type of metaplasia that looks more homogeneous. It still has little goblet cells and stuff, but is different from this set of metaplasia, which this I'll show at the end is more of the high-risk thing. And just to put this in order, here's inflammation. Over here was a gastric cancer. So these are probably a precursor lesion over to that. So just to kind of go into, not all metaplasias are the same. And this is important because this, I think we should figure out, if we can diagnose this, we can tell who needs to be surveyed and who probably can be exited from surveillance. So this is a very complicated slide. I'll go into it very slowly, but I'll just, this essentially shows that there are 3 types of metaplasia. Complete, which is the low risk type. Then it switches to incomplete, which is a much higher risk. So I'll kind of go over the features of these and I'll show pictures and I'll explain it very carefully. I think you'll get it by the end. So complete, you generally have organized glands. They're nice linear, they look calm, they're not wild appearing. The goblet cells all look the same. These are the mucus secreting cells that actually, I'll show that are the big feature of intestinal metaplasia. They shouldn't be in the stomach normally. And then you have the absorptive cells that have a nice brush border. This is what absorbs all the nutrients in the small bowel. And then you have some Panna cells, which are bottom of the crypts. But these are all features of the small bowel. So the small bowel is a very low risk for ever developing cancer. We don't offer surveillance to look at the small bowel, the region between the stomach and the colon. However, everybody in the United States, everybody over the age of 45 gets offered a colonoscopy. The reason is colons develop cancer. And just the same as here, this incomplete type metaplasia mimics the colon. It has goblet cells of variable size. The absorptive cells start getting these mucins in it that they shouldn't have. And there's very few Panna cells. So essentially, this is the low-risk type. Looks like the small bowel, and just like the small bowel doesn't get cancer very often, this confers about a 4 times increased risk and looks like the colon. The problem is discriminating between these 2 types because they look the same with normal stains. You actually need special stains to figure out if they have type 2 or the very high-risk type 3 things. And to do this, you actually have to look at the type of mucins they make. So type 2 is characterized by salic acid. This is a sugar that's got an acidic group on it. And then that acidic sugar switches to a sulfate, which is what confers the highest risk here. And I'll show you that this is a common feature of all foregut metaplasias, that the switch to a sulfation is actually an incredibly important thing and puts you at much higher risk to develop cancer. So what does this actually look like? So this is the low risk type. This is the one that looks like the small bowel. You have a lot of homogeneous blue cells here. So blue is staining mucins that are salic acid, essentially. So you see all these are this, but you see the other cells in between them don't have any of these mucins. This is what changes in incomplete metaplasia, where you see all these cells around you. You still have these little round goblet cells, the mucus ones, but all the other cells start to express these mucins. This is type 2, this is intermediate risk. What the switch is to high risk is when you start expressing sulfation, which is done by this brown dye here, high iron diamine, which binds sulfate. But you can see that the blue is largely switched to this brown pigment here. This is the highest risk. And this is not just in the stomach that changes its risk. Throughout the esophagus, stomach, and pancreas, we have found that this switch is what really makes the highest risk to develop cancer. And this is the population we need to identify so that we can survey them and hopefully catch cancer at a very early stage when it's curable. And so, the one problem is we no longer actually have a way to identify this. Because diamine is incredibly toxic. It has been taken out of all commercial laboratories. So right now we can say whether it looks like the small bowel or whether it looks like the colon, which is higher risk, but we can't say whether they have the last step right before dysplasia in cancer. So we, in my lab and also in collaboration with Kosik Das, we have an antibody that actually we believe now can do this. And that's obviously a non-toxic solution. And I'll show you that it's not, pretty much all these foregut metaplasias express this right before they develop cancer, essentially. Barrett's esophagus is 95% sensitive and specific. Pancreatic, whether it's a cyst or that, it's incredibly sensitive right at the highest grade things. The one thing is, this antibody, before we knew what it bound, it didn't look like it worked very well in the stomach. But the reason is now that we know that it exclusively binds sulfate and will not bind sialic acid, we're now testing whether this 35% here, the 65% we missed, is because that was type 2 and not type 3, not the high risk. This should only detect the high risk metaplasias. So the reason I bring this up is that if you look at this esophagus, stomach, and pancreas, if you have these sulfated mucins expressed, we survey all these people every 2 years. That way we can find when they get high-grade dysplasia and ablate them. Pancreas, we've actually run a phase 2 multi-center clinical trial showing that if you express these sulfated mucins, you should just have your pancreas taken out. You have essentially either carcinoma in situ, which is cancer that hasn't spread yet, or you have high-grade dysplasia. However, stomachs, we just sit on this because A, we don't have a commercially available test because diamine's toxic has been taken out of all commercial laboratories. And B, I don't think surveillance, we don't do as much as we really should be because there's a lot of reasons, but I think we can do better. And the other thing is I'm not showing something profoundly brand new. Here's an immunofluorescence from 1968 showing the expression of sulfated mucins right next to gastric cancer. It's just that this has been largely ignored. It's in the guidelines as, what the highest risk, but we don't test for it. We have no way to test for it commercially. So hopefully, this is part of our PREVENT trial. One of the aims is actually pretty much validating that DOS1 antibody is a non-toxic alternative to identify the highest risk people that should be surveyed to find gastric cancer at a time where it's curable and not when it's too late. Versus now, the pathologists will say there's metaplasia, but there's nothing we can do because insurance companies won't fund surveillance or anything like that. But if we can say, this is not just metaplasia, this is type 3 intestinal metaplasia, the one that is the highest risk, hopefully we can get those people screened or surveyed. And the other thing is we also might now have treatments. So Jim Goldenring's lab, this was just published like a month or two ago, used a MEK inhibitor and is actually able to downgrade the histological grade of intestinal metaplasia. Because I said before at the beginning that once it gets metaplasia, you have replaced normal stomach cells with cells that have no business being there that should be in the intestine. This is kind of generally believed to be a permanent transformation. With this, they've actually been able to downgrade this histology and get rid of some of the metaplasia. So potentially, the ones with the highest risk, this is a phase 1 trial. They haven't correlated this with patient outcomes or anything. They've just shown that under the microscope, these stomachs look better with just 2 weeks of MEK inhibitor. And that's pretty much what I got here. I'd like to thank my lab who do everything, Shabo, Shwime, and Gabe. Collaborators, Kosik Das, who brought this original antibody to me, and I do all my translational work with him. Kay Lee, who's a pathologist that I work, she's at Hopkins now, moving to SLU pretty soon, and she is what I'm trying to validate the antibody and higher diamine with, and funding, and thank you all for your attention.
So now I'd like to introduce our last and final speaker of the session, Dr. Vladimir Kushnir. He is professor of medicine, Division of Gastroenterology. He is director of gastric endoscopy, interventional endoscopy, and the interventional endoscopy fellowship program. So please welcome Dr. Kushnir.
Thank you, and thanks for having me. So this is going to be a complete 180. Number one, we're going to talk, this is going to be a very clinically oriented talk. And number two, we're really going to be talking mainly about the esophagus and specifically Barrett's esophagus. And I think a lot of the concepts that Dr. Brown introduced as far as intestinal metaplasia and progression of that to dysplasia and cancer apply here as well. So we'll talk about the basics of Barrett's esophagus, then discuss what endoscopic therapy options we have today, and some of these do apply to early gastric cancer as well. And then we'll talk about who to treat and when. So basics of Barrett's. So Barrett's is intestinal metaplasia of the esophagus. So just like in the stomach, when you have something chronically irritating the esophagus, you can trigger molecular pathways that make the lining change from squamous mucosa, which is basically just like your skin, to columnar mucosa, which looks like the small intestine and has goblet cells, which are these little mucus-producing cells, which in somebody who has chronic reflux can actually be protective and decrease the risk of them getting esophagitis, strictures, and other complications. So if you live to the age of 40, Barrett's esophagus may be a helpful adaptive mechanism. If you live to the age of 70, though, accumulation of that intestinal metaplasia being exposed to gastric acid and other toxins can progress to esophageal cancer. So why should we care or treat Barrett's esophagus? Well, about up to 7% of the U.S. population is affected by Barrett's. This has increased recently because of increase in processed food, increase in obesity and reflux. And we know that Barrett's is really the only precursor to esophageal adenocarcinoma and increases the risk 30 to 40-fold over the general population. And we also know that if we get rid of it before it turns to cancer, we can prevent esophageal cancer. So that's a total win. And unlike gastric cancer, esophageal cancer has been going up very rapidly, especially adenocarcinoma, over the past several decades. Again, the hypothesis is that it's largely driven by the obesity epidemic, as well as consumption of processed foods and just changes in lifestyle. So esophageal cancer is a major problem, and unfortunately, despite advancements in chemotherapy and surgery, we really haven't budged survival much. So how do we find Barrett's esophagus? Well, in 2025, still the way to screen for Barrett's esophagus is doing an endoscopy, obviously resource intensive, and not everyone can have one. But when we do an endoscopy, Barrett's esophagus looks like this. So it's this, does the pointer work? So it's this pink, this is the top bottom of the stomach, so you can see the folds that you saw in all the pictures earlier. This is the esophageal lining, that tissue that's basically like the skin on your hands. And this is Barrett's, so it looks like these pink salmon-colored tons coming up into the esophagus. So this is that intestinal metaplasia. So this is what we look for, and this is how we know there's Barrett's esophagus. How do we figure out if it's bad Barrett's or benign Barrett's? We take biopsies. The traditional protocol is we just take a lot of pinch biopsies throughout, and the more you look and the more biopsies you take, the more likely you are to find dysplasia, and again, more likely you are to find something you can treat and to prevent cancer. These are the things that you can find on biopsy. So just for reference, this is what the normal esophagus looks like. This is Barrett's. So again, you see that intestinal metaplasia. You can see a very different structure of the cells here. You can see these little goblet cells. That's what really defines intestinal metaplasia in the esophagus. And then it can start going toward dysplasia. So this is low-grade dysplasia, maybe something to worry about. These cells are starting to look a little irregular. You can see here dark, kind of these there's more purple. Generally on biopsies, the more purple you see, the worse it is. And here you see a little more purple. But low-grade dysplasia can also be seen in inflammation in the esophagus and not diagnostic of something that will progress to cancer. Then you have high-grade dysplasia, which is basically, which in Asia, for example, is referred to as carcinoma in situ. This is cells growing out of control, glands forming where they shouldn't, but they're limited to the superficial lining of the esophagus. So high-grade dysplasia, if we can just get rid of the top lining of the esophagus that faces the food, we can treat it and prevent cancer. And finally, you get invasive adenocarcinoma, again, irregular growth of glands where they shouldn't be. So what is the risk of Barrett's progressing to esophageal cancer? Well, the good news is non-dysplastic Barrett's, which is what about 90% of the people with Barrett's esophagus are diagnosed with, has less than 0.5% per year chance of progressing to cancer. Nobody would really argue that we need to treat that because, frankly, your chance of dying in a car accident is probably about equal to that. Low-grade dysplasia is a more controversial area. Studies are kind of all over the place still. The risk of progression is anywhere from 0.5% to 7% per year to high-grade dysplasia or cancer. And finally, high-grade dysplasia, there's really no argument about treating. The risk of progressing to esophageal adenocarcinoma is at least 6% per year. So in somebody who is at all fit, it's something that should be treated with ablation. So what can we do with medical management to prevent Barrett's from progressing to cancer? Well, anti-reflux surgery is great for treating symptoms, but doing a surgery to treat reflux does not decrease your risk of esophageal cancer. Medications may help. So aspirin and other nonsteroidal anti-inflammatory medications may slightly decrease the risk of progression to esophageal adenocarcinoma in someone with Barrett's. But they're not currently recommended solely for esophageal cancer prevention because of the risk of complications. And finally, esomeprazole. So proton pump inhibitors are very effective at decreasing risk of progression to esophageal adenocarcinoma and very safe medication. So anyone with Barrett's esophagus really should be on a proton pump inhibitor because that decreases the exposure to gastric juice and acid and decreases the risk of the Barrett's progressing to cancer. So how can we treat it? Well, again, Barrett's is a mucosal disease. It affects just the surface lining of the esophagus. So it's perfect for us doing something with a flexible endoscope. So it only goes 1 to 4 millimeters in depth and is not found below the muscularis mucosa. So it's in just the very, very lining of the esophagus. So we can treat it just like we treat a colon polyp by removing it. Except Barrett's usually is flat and involves a larger area, so cutting it all out is not practical and frankly fairly risky. So what do we do to treat it? What else can we treat? So we can also treat early esophageal cancer. So we know that the risk of lymph node metastasis in esophageal cancer that's just limited to the superficial lining is basically 0. It's less than 2% if it's limited to kind of the top, if it doesn't go into the submucosa. But once it gets into the submucosa, so past the superficial lining of the esophagus into toward the lymphatic vessels, the risk is greater than 20%. So we can treat early esophageal cancer endoscopically. This also applies to early gastric cancer. We can treat early gastric cancer that hasn't gotten below this muscularis mucosa level. We can treat that endoscopically and avoid surgery in a lot of patients. So to remove Barrett's with larger areas that don't have any nodules or nothing that looks too worrisome, we can basically burn it off. And if it heals in a non-acid environment, it reverts back to normal lining. The standard approach that we use here is radiofrequency ablation. It's a balloon or a catheter. You can see here this electrical coil on it. It inflates and you apply an electrical current to the esophagus and you remove the bad tissue, and then once it heals, it basically turns back to a normal esophagus. So this is what the procedure looks like. You have an endoscopy. We place this catheter in, inflate it, burn the esophagus. Most people have some chest pain because burning your esophagus is not going to feel like a bed of roses after. And then it goes from looking like this, so you can see very extensive Barrett's esophagus, to normal esophageal lining. This procedure was evaluated now 15 years ago in a very large multi-center randomized controlled trial, so the highest level of evidence that we have, and definitively proved that ablating the Barrett's, number one, you can get rid of it with endoscopic therapy, but more importantly, you can dramatically decrease the risk of progression. So you can see for high-grade dysplasia in the study over 2 years, 19% of patients progressed to cancer versus just 2.4% of the patients who were treated with radiofrequency ablation. So very effective treatment to decrease risk of cancer in patients with dysplastic Barrett's. Overall, very safe. Most patients have chest pain after, but strictures, so scar tissue in the esophagus that makes it hard to swallow, are the most common complication, happening in about 6%. More serious complications like bleeding and chest pain requiring hospitalization can happen, but are exceedingly rare. Now, treating Barrett's can be challenging because, like anything, it's not everything's created equal. So patients with a lot of Barrett's, so more than 3 centimeters, typically very difficult to treat endoscopically. We just have to burn it over and over and over again to get it all to go away. So it requires more ablation and obviously more burdensome for the patient. More importantly, these patients who have masses or nodules in the esophagus. So these are always, you have to make sure that this is not cancer. You can't just burn over this. You have to remove it and send it to a pathologist to make sure that it's not growing too deep. So you have to inspect the esophagus very carefully, and if you find something like this, you have to resect it, and I'll go over the endoscopic resection techniques in a second here, and then you can ablate the rest of the Barrett's esophagus. So how do we resect it? Well, the most common technique is something called endoscopic mucosal resection. Basically, we try and do what we do during a colonoscopy and remove these nodules with a loop snare. Today, we mainly do it by deploying a rubber band, so you can think of kind of how they tie off a little extra finger when a baby's born with it, or how they sterilize animals on a farm. You put a rubber band around it, and then it makes it a polyp, and then you can chop it off with a loop snare. So this is what it looks like in a video. So you can see here a patient with Barrett's. And we're going to come up on, and there's a nodule right there. So you can see the subtle raised area that's worrisome for a little bit of cancer. And then we deploy a rubber band. And you can see the loop snare coming out here. And you can see resected here. And you basically go, the other technique is you can lift it with a little bit of blue water. And again, remove the tissue with a loop snare. And you can see here resecting a very large area of normal esophageal tissue, getting some more there. And this allows us to send the tissue for a pathologic examination to make sure it's not growing into the submucosa, make sure we're appropriately risk stratifying the patient so that if they do have invasive disease, they go for surgery or chemotherapy and radiation. The other technique that we use, and this is used in gastric cancer as well, is endoscopic submucosal dissection. So this is a more complex method where we actually peel the polyp or the mass off of the lining of the stomach. Again, what we do is we lift it with a little bit of salt water and then use an endoscopic knife to cut the tissue off. And this allows us to remove larger pieces of tissue than what you can put a rubber band around. So you can take 3, 4, 5 centimeters tumors off with this technique. This is an example of doing this with an esophageal squamous cell cancer. So you can see here there's a mass in the esophagus. We mark it out with electrocautery, and then you can see lifting it with a little bit of blue water. And then using the electrosurgical knife to basically cut it, peel it off. Kind of the way I describe it to patients is it's like we're peeling an orange. We're taking the peel off of the orange and leaving everything underneath. So you can see here dissecting. This is a submucosal tissue, connective tissue. And the goal is obviously to stay away from the muscle layer and not go all the way through the wall of the esophagus or the stomach. And now you can see here, there's the final product. So both of these techniques are usually combined with ablation for Barrett's esophagus because you need to get rid of all the Barrett's. These techniques, the main differences are, number one, EMR is much easier than ESD and takes a lot less time. But with ESD, you can get en bloc resection, so removing the whole tumor in one piece 90% of the time. And you can get an R0 resection, meaning clean margins, over 70% of the time. Complication rates with ESD are a bit higher because it's a longer, more complex procedure, but overall both very safe and much safer than surgery, as we'll see in a minute. Other things that you can do for dysplastic Barrett's is freeze it, so similar to what dermatologists do for skin cancer or OBGYNs do for early cervical cancer. We have several techniques for actually freezing Barrett's esophagus. One of them is a cryoballoon, so it's a little balloon that you inflate in the esophagus, and you can see a little bit of Barrett's right there. And we freeze it with nitrous oxide, so similar to what you do to a wart. And this leads to, again, the tissue sloughing off and getting replaced with normal esophageal lining. And this actually compares pretty well to radiofrequency ablation. The other technique that we can use for treating Barrett's, but also for palliating esophageal cancer, is spray cryotherapy. So this is a different way of delivering cold. So this is liquid nitrogen, and this is actually sprayed through the scope. So this gets very cold. The endoscope actually freezes as you do this. And this can penetrate very deeply, so we use it sometimes for esophageal cancers in patients who can't swallow to debulk it once they've exhausted their options with radiation. And spray cryotherapy works pretty well. It palliates patients with esophageal cancer and allows a lot of them to avoid a stent or avoid a feeding tube for quite some time. So how are the long-term outcomes with endoscopic treatment for Barrett's? Well, in short, very good. About 90, 95% of patients remain in remission years later. But the Barrett's can come back. So the rate of recurrent high-grade dysplasia or cancer after we endoscopically eradicate the Barrett's is about 5%. So that means these patients are coming for annual endoscopies kind of as long as they're healthy enough to have a scope. So last part of my talk is who do we treat and when. So again, we go back to the slide of kind of the risk of cancer progression. Low-grade dysplasia is the controversial area, and that's because inflammation can look like low-grade dysplasia. So 85% of patients who are initially diagnosed with low-grade dysplasia on a biopsy are reverted to non-dysplastic Barrett's and have a very low risk of progression. But those, when an expert pathologist looks at it again, says, yes, there's low-grade dysplasia, are at high risk for progression and should be treated. And here, kind of similar data, so you can see ablation of low-grade dysplasia that's confirmed leads to 11% absolute reduction in progression to high-grade dysplasia or cancer. The other more interesting area, and the area that makes me lose sleep at night, is what to do with early esophageal cancer that invades the submucosa. So we know that esophageal cancer, and this applies to gastric cancer as well, once it gets deeply into the submucosa, so toward the muscle layer of the GI tract, the risk of it spreading to lymph nodes goes up significantly, 25% or more. And we also know that poorly differentiated tumors, larger tumors, and deeper submucosal invasion increase that risk. However, we also know that superficial submucosal invasion probably has a very low risk of lymph node metastasis. So this is where getting an en bloc resection with ESD is very important, and very careful pathologic examination is important when we're seeing these patients, because nobody wants to have their stomach removed and even worse than that, have your esophagus removed. Because both of those are very morbid operations. And if we can save even a few patients from having that surgery with endoscopic therapy, it is very beneficial. So this is why ESD is really going to be becoming more and more of the technique for removing these early lesions. And just comparing outcomes to surgery for esophageal cancer. So for T1A, this is limited to the very superficial lining of the esophagus. You can see endoscopic therapy is much fewer complications, equal survival, much shorter hospital stay, if at all. A lot of these patients can have an outpatient procedure. For T1B esophageal cancer, so again, the data that we have is that endoscopic resection is actually pretty close to esophagectomy for outcomes. Now, these are very carefully selected patients, but surgery for deeply invasive esophageal cancer doesn't have the best outcomes in times, because these patients tend to recur because of the anatomic location of the esophagus. So endoscopic therapy for early esophageal cancer and early gastric cancer is something that we're definitely doing more and more of, and I think is going to grow into the future, and makes early detection that was talked about earlier in the prior talks very important, because if we can detect these things early, we can either prevent them or resect them endoscopically and avoid morbid surgeries like gastrectomy or esophagectomy. Thank you.
So would all the speakers from the first session be able to stick around, come up front, and we can take maybe 15, 20 minutes of questions, or is that okay? So I'll ask the first question, and I guess it goes to everyone. So as our screening techniques kind of improve for other GI cancers, like I'll just use colon cancer as an example, we're doing more sophisticated, I guess, blood tests, trying to detect circulating tumor cells, circulating tumor DNA. What progress have we made in kind of these new technologies in upper GI cancers, and especially for these, hereditary diffuse, they tend to be kind of, I guess, freely moving. So, maybe not just in the blood, gastric juices. Can you guys speak on that?
I can start with esophagus.
Oh, okay, go ahead,
So
Vlad.
esophageal cancer, so screening uptake has still not been that high, because for esophageal cancer screening for Barrett's, you kind of have to have an upper endoscopy. And that's been the paradigm up until now. However, today we do have several technologies that are now FDA approved and rolling out, which are actually little capsules that are attached to a string that you swallow and then get dragged back out. And we look for either methylation signatures or basically cytological markers of Barrett's esophagus and esophageal cancer. And this is something that is much lower cost than endoscopy and also can be done at a primary care doctor's office or on a screening van like we do for prostate cancer or breast cancer screening. So potentially is going to really broaden the number of patients who we can, in a cost-effective way, screen for Barrett's and by inference esophageal cancer. And that's something that, you know, is here now, although not widely adopted.
For HDGC, really no progress in that area. I think the lesions are too small for circulating tumor DNA to be affected. There is some interest in that approach of trying to sample the mucosa directly, but you know, it's multifocal disease. It's really tough to do that thoroughly. So unfortunately, for now, we haven't really got progress with that approach.
I'll just say, I'm not aware of much progress on the side of stomach cancer related, not related to hereditary, but what I would say is that H. pylori screening is something that I think is still poorly done. We do a very bad job of screening for it. As gastroenterologists, it's almost embarrassing. And we do a very bad job of making sure that when we do treat it, that it's been eradicated. And there have been multiple studies showing that actually primary care physicians do a better job of managing or detecting eradication than gastroenterologists. And so I think it's, just to quote David Graham, who was sort of the guru of H. pylori and is at Baylor, he often said that the problem with H. pylori is that we treat it as a gastric disease, not as an infectious disease. And as a result, gastroenterologists managed it and not infectious disease doctors. So I think we probably need to do a better job of screening and getting rid of it when we do detect it.
Yeah, and I'll just make a comment. Well, I'll talk about it later. But yeah, so like I said in my last slide, is that at least the stuff I'm studying involving these sulfate abuses, this is not something new. I think it's just a consequence of just not having adequate funding for gastric cancer awareness and just, you know, I mean, I'm pursuing something that was known in 1968. I just have an easier way to detect it now and a way we can take a high throughput in any histological lab we do it. It's just that I think the funding is holding us back, and we don't have as much awareness and everything as for the stuff we do. We know what to do, and we just need to do it. So.
There's a Zoom question, so we'll pull it up on the screen, and you guys can, we'll just have you guys answer that. I'll read it once we get it put up on the screen here. Sorry, I guess you can unmute yourself. Volume. No volume. You want to just type it real quick? Sorry. DJ, can we see the chat as well? Is that okay? I don't think. So yeah, sorry, I mean, I guess please just type into the chat message, and Dr. Guilford is just, I'll read it, or, I mean, everybody can see it, so.
Okay, a question about potential gene therapy. I look forward to hearing, Gavin, what that might be. I mean, you know, obviously gene therapy is the way to ultimately make this problem go away with HTGC. As far as I'm aware, it's still in its infancy, and so nothing is really even close, but I'd love to hear what Gavin's been doing in that area, so I'll look forward to your email, Gavin.
So I guess oh. Dr. Caffey.
Thank you for the talk. My name is José. I'm a thoracic surgeon at the University of Missouri, and thank you, Dr. Guilford. My question is for you. You elaborated very well on the discussion with patients on the prophylactic gastrectomies and how challenging that is, I can imagine. How do you find in invasive cancer? Is there any data on the prophylactic gastrectomies, on the specimens? I mean, how many patients get upstage, basically?
Very, very rarely. Very, very rarely. People have prophylactic gastrectomies. I mean, it's almost unheard of. If they have their surgery in their 20s, very, very rarely is it upstaged. Just
Thank you.
Thanks for all these nice talks. Dr. Guilford, I had a question for you. With the local therapy that you're talking about, the techniques that you show wouldn't differentiate between the drug that binds to the surface of the cells and the drug that gets into the tissue. Do you know how deep the drug has to get? Do you know that your drug can be absorbed superficially and that you can tell that difference?
Yeah, we don't need to get deep. It's only a matter of a few millimeters, and we've looked with one drug so far, and that was a drug called dasatinib, and that did get down to where we would need it to get to. So I guess it will vary from drug to drug, but certainly, if the drugs have the right charge structure, then they do get into the stomach and can diffuse into the right depth. So there's some, I can't give you the details of the charge structure, but our chemists are very careful with which drugs they consider to be ones which are amenable to this approach.
Well, if they do get absorbed, then what doesn't get absorbed in the stomach, because the residence time in the stomach will be relatively short, then it's going to be absorbed in the small bowel.
No, but it won't. I mean, most drug will be taken and will be released over a period of hours into the stomach surface. But any drug which remains bound to the mucin and goes into the bowel is inactive. So the drug is inactive while it's still bound to the mucin, to the mucoadhesive compound.
Everything that gets acid released will stay in the stomach.
Oh, no, some will go, will go, but I think we'll be able to tune that process a little bit by, we can tune the rate of release of drug from the mucoadhesive polymer. That can be tuned, and so it can come off over whatever number of hours we want it to happen to reduce the likelihood of it just being released into the stomach as a whole and lost into the bowel. So we won't get perfect uptake only in the stomach, but the one experiment we've done so far in a mouse study was we had a 70% reduction in dasatinib and other organs compared to the stomach.
So the PK is complicated, but it sounds like you're in the right direction.
Yeah, right, exactly, yeah.
Okay, I think we'll close out the first session, so let's give the speakers a round of applause. We can take a 10-minute coffee break and just meet back here. Thank you.
Zoom audio check. Zoom audio check. Audio check. Zoom audio check.
Someone in the family was tied in for like many years ago. He wasn't really a family member, he was an in-law to the family. But it was good, so I really like the family. Oh, you're a Dolphin fan? Yeah, oh, they beat the Vikings in the Super Bowl. One of 4 losses, sad to say. Having issues with one quarterback's been injured. Now they have Carson Wentz that plays the Eagles.
Zoom audio check. Zoom audio check.
Eagles have 2 losses in a row, they're gonna come out aggravated. That may be a tough one, but the Vikings have defensively stuff. Eagles, they come apart.
Nice to meet you. So we have first line, HER2 positive shield, and of course, well, 2 things. One is, so mother-in-law had when did the treatment course. I don't think she completed it, still has it.
Side effects. Typically now, the treatment is you take 4 different medications. You take a PPI that acid. You take this one, which is a small molecule itself causes some kind of upset stomach. And you take a couple of pills. You have to treat that up to 4 times a day for 2 weeks straight. And what happens is a lot of patients, they take it, they cause upset stomach, they miss doses, they don't finish their medications for whatever reason. And then it's really important, that's why we need to follow up with a lot of it. Finished a month later, we need to retest them, either in stool or sometimes we do breath tests, looking for H pattern to see if it's still there. If it's still there, there's different reasons. One is if you didn't complete the treatment, you can't get the same treatment again, but this time making sure that you complete it. But 2, sometimes there's resistance. So these bugs are resistant to the different antibiotics.
So she may have completed the course.
If she completed it and she's still positive, then there's other therapies. So we do what's called salvage therapies, where we do other treatment regimens to make sure that we overcome that resistance. Usually that's the reason why.
I just assumed she completed, but now that you mentioned data.
It's possible. So now there's ways, they're usually set up where you can set off, for example,